Welcome, everyone. Today, we're diving into an exciting journey with our biology viral quiz. This is the first slide, the cover slide, so I want to warmly welcome you all. We'll be exploring the fascinating world of viruses and biology through engaging questions and quick thinking. The title of our presentation is Biology Viral Quiz, and you'll see a fun question, think, answer format throughout. In this session, we'll cover key concepts about viruses, how they operate, and test your knowledge along the way. So, get ready for an interactive and informative experience. So, let's explore the question, powerhouse of the cell. Now, many of you might already be thinking about which organelle this refers to. Well, the answer is the mitochondria. These tiny structures are essential because they generate the energy cells need to function. Think of them like the power plants of the cell. They take in nutrients, like glucose, and convert them into a form of energy called ATP, which powers everything from muscle contractions to nerve signals. Understanding the role of mitochondria is crucial because they're not just about energy. They also play roles in cell signaling, growth, and even programmed cell death. This makes them vital for maintaining healthy cell function and overall organism health. So, as we continue, keep in mind how central these organelles are to the cell's energy management and overall vitality. They truly are the powerhouse, enabling all the other cellular processes to happen smoothly. Answer coming. 5 seconds think. Now, take a moment to consider what this slide is really about. We're focusing on the idea that sometimes answers aren't immediately clear and require a bit of reflection. It's about giving ourselves the space to think deeply before jumping to conclusions. This is crucial, especially in the context of our overall theme, which is about making informed and thoughtful decisions. By pausing and contemplating, we improve our understanding and make better choices moving forward. So remember, taking that extra moment can make all the difference. Let's keep that in mind as we continue. Let's take a closer look at mitochondria, often called the powerhouses of the cell. These tiny organelles are essential because they generate most of the cell's energy through a process called oxidative phosphorylation. In simple terms, they convert nutrients into usable energy in the form of ATP, which powers nearly all cellular activities. What makes mitochondria particularly fascinating is their unique structure. They have a double membrane, with an inner membrane that folds into cristae, increasing surface area for energy production. Interestingly, mitochondria also contain their own DNA, which means they can produce some of their proteins independently of the cell's nucleus. This self-sufficient nature hints at their evolutionary origins. They are believed to have originated from ancient bacteria that formed a symbiotic relationship with early eukaryotic cells. Understanding mitochondria is crucial because their dysfunction is linked to a range of diseases, from metabolic disorders to neurodegenerative conditions. So, in the context of overall cell health and energy metabolism, mitochondria truly are central players. So let's explore the idea of green pigment in plants. Why are plants green? Well, the key pigment responsible for their green color is chlorophyll. It's essential for photosynthesis, which is how plants convert sunlight into energy. This pigment absorbs mainly blue and red light, but it reflects green, which is why we see plants as green. Understanding this pigment is crucial because it ties directly into how plants make their food and sustain themselves. It also highlights the connection between plant biology and the broader theme of how life harnesses energy from the environment. So, in essence, this simple color tells us a lot about the fundamental processes that keep plants, and ultimately us, alive. Answer coming. Hourglass not done. 5 seconds to think. In this moment, I want you to pause and really consider the question at hand. Take this time to reflect on what we've discussed so far and how it connects to the bigger picture. Sometimes, giving ourselves a moment to think leads to clearer insights and better decisions. Remember, this pause isn't just about waiting. It's about engaging more deeply with the content and preparing for the next steps. So, use this brief moment wisely, and let's continue onward once you're ready. chlorophyll is the green pigment that plays a crucial role in photosynthesis, which is the process plants use to convert sunlight into energy. It's what gives plants their vibrant green color and allows them to absorb light efficiently. Specifically, chlorophyll absorbs light most effectively in the blue and red wavelengths, while reflecting green, which is why plants look green to our eyes. Understanding chlorophyll helps us appreciate how plants harness sunlight, supporting not just their growth, but also the entire ecosystem by producing oxygen and organic compounds. So, in the context of our presentation, chlorophyll is central because it underpins the fundamental process that sustains life on Earth through photosynthesis. So the question here is, what is the basic unit of life? This is a fundamental question in biology, and understanding it helps us grasp how living organisms are built and function. Essentially, the basic unit of life refers to the smallest structure that can carry out all the processes necessary for life. And, as we'll see, that unit is the cell. Cells are the building blocks of all living things, from tiny bacteria to complex humans. Recognizing this helps us connect the dots between the structure and function of living organisms, and it's a key theme in understanding biology as a whole. Answer coming. 5 seconds think. All right, take a moment to gather your thoughts. This slide is a bit of a pause, giving us time to reflect. It's important to remember that sometimes the best insights come when we allow ourselves a few seconds to think. So, as we wait for the answer, think about how this concept ties into the bigger picture we've been discussing. It's all about encouraging a thoughtful approach, not just rushing to conclusions. Now, let's see what the answer is, and how it reinforces our overall theme of deliberate and strategic decision-making. Let's talk about the concept of a cell. When we refer to a cell in this context, we're talking about the basic building block of life, but it also plays a crucial role in various scientific and technological fields. Now, the key point here is that understanding cells allows us to grasp how living organisms function at a fundamental level. This knowledge is essential because it connects to many broader themes, such as biological processes, medical advancements, and even innovations in biotechnology. So, when I ask you to think about a cell, I want you to see it not just as a tiny unit, but as a powerful foundation that influences many aspects of science and health. And this understanding ultimately helps us innovate and improve our approaches across different disciplines. So let's move on to our next question. What is the genetic material in most organisms? This is a fundamental question because understanding what carries genetic information helps us grasp how traits are inherited and how organisms function at a biological level. Most organisms, including humans, plants, and many microorganisms, store their genetic information primarily in the form of DNA. DNA, or deoxyribonucleic acid, is a long, double-helix molecule that contains the instructions necessary for building and maintaining the organism. It's important to highlight that DNA is incredibly efficient at storing vast amounts of information in a compact form. This genetic material is passed from generation to generation, ensuring continuity of traits and characteristics. While some viruses use RNA instead of DNA to store their genetic information, in the context of most living organisms, like those we're discussing today, DNA is the primary genetic material. Connecting this back to our overall theme, understanding the nature of genetic material helps us appreciate the molecular basis of heredity and the mechanisms underlying evolution and diversity among living organisms. Answer coming. Hourglass not done. 5 seconds to think. Now, let's take a moment to reflect on this. This slide is all about patience and the importance of taking time to consider our responses. Sometimes, we're tempted to rush through decisions or jump to conclusions, but pausing can make a huge difference. It allows us to gather our thoughts, analyze the situation more clearly, and make better choices. This idea ties directly into our overall theme of mindful decision-making and strategic thinking. Remember, giving ourselves that extra moment can lead to more effective outcomes and stronger results. So next time you're faced with a tough question or a complex problem, just pause for a moment, like now, and really think it through. Let's take a closer look at DNA. As many of you know, DNA stands for deoxyribonucleic acid, and it's essentially the blueprint of life. It contains all the genetic information necessary for an organism's growth, development, and functioning. What makes DNA particularly fascinating is its structure. It's a double helix, like a twisted ladder composed of nucleotide pairs. These pairs are made up of 4 different bases: adenine, thymine, cytosine, and guanine. The specific sequence of these bases encodes the instructions our cells need. Understanding DNA is crucial because it underpins fields like genetics, medicine, and biotechnology. It connects directly to how traits are inherited and how genetic information is passed down through generations. So, in essence, DNA is not just a molecule, it's the fundamental code that defines life itself. Let's explore how plants make their food. This process is fascinating because it shows how they harness energy from the environment. Basically, plants take in sunlight, which is a vital source of energy. They also absorb carbon dioxide from the air and water from the soil. Using these ingredients, they perform a process called photosynthesis. During photosynthesis, plants convert sunlight into chemical energy, producing glucose, which is their food, and oxygen as a byproduct. This process is essential not only for the plant's survival, but also for supporting life on Earth, since it provides oxygen and forms the base of most food chains. Understanding this process helps us appreciate the incredible way plants sustain themselves and contribute to the environment. It's a perfect example of nature's efficiency and interconnectedness, which ties into our overall theme of how living organisms interact with their surroundings. Answer coming. 5 seconds think. Let's take a moment to reflect on what we've just discussed. This slide is all about pausing and giving ourselves a moment to consider the key points. In many situations, especially when making decisions or solving problems, a brief pause can make a huge difference. It helps us process information more clearly and avoid jumping to conclusions. By thinking intentionally for just a few seconds, we can improve our understanding and make more thoughtful choices. This practice connects directly to the overall theme of mindful decision-making and strategic thinking. So, let's embrace these short moments of reflection as a vital part of our process. Let's take a closer look at photosynthesis. This process is fundamental to life on Earth because it's how plants, algae, and some bacteria convert light energy into chemical energy. Essentially, they take in carbon dioxide from the air and water from the soil, and using sunlight, they produce glucose and oxygen. The glucose serves as food for the plant, providing energy for growth and development, while the oxygen is released into the atmosphere. What's really fascinating here is how this process links to the bigger picture of life on Earth. It's the foundation of most food chains and helps regulate atmospheric oxygen levels. Photosynthesis isn't just about plants making food. It's about creating the oxygen we breathe and maintaining the balance of gases in our environment. So, understanding this process helps us appreciate the vital role that green plants play in supporting life as we know it. So let's explore the question: gas released during photosynthesis. Now, this is an interesting point because photosynthesis is primarily known for taking in carbon dioxide and releasing oxygen. But in some cases, plants and other photosynthetic organisms can also produce and release other gases. For example, they might emit small amounts of methane or other volatile organic compounds, especially under certain environmental conditions. However, the most significant gas associated with photosynthesis is oxygen, which is a vital byproduct that supports life on Earth. Understanding this helps link the process of photosynthesis to the larger picture of how energy flows and gases cycle through our planet's ecosystems. So, while oxygen release is the main gas, it's also important to recognize that other gases can sometimes be part of this process, depending on the organism and conditions. Answer coming. 5 seconds think. All right, take a moment to think about this. This slide is all about pausing and reflecting before we move forward. It's a reminder that sometimes the best insights come when we take a brief pause. By giving ourselves a moment to consider, we can approach the next steps more thoughtfully. This concept ties into the overall theme of strategic thinking and deliberate decision-making. So, let's keep in mind that a little pause can lead to clearer, more effective actions. Let's take a closer look at oxygen. Oxygen is essential for life as we know it. It plays a critical role in processes like respiration, where our bodies convert nutrients into energy. Without oxygen, our cells can't function properly, and survival becomes impossible. In many environments, oxygen levels can vary, which impacts how organisms adapt. For example, some species have developed ways to survive in low oxygen conditions, while others rely on abundant oxygen to thrive. Understanding oxygen's role helps us appreciate its importance not just biologically, but also environmentally. It ties into the broader themes of sustainability and the balance of ecosystems. So, when we talk about oxygen, we're really discussing a fundamental element that connects health, ecology, and the stability of life on Earth. All right, let's move on to our next question. What is the organelle containing chlorophyll? Now, chlorophyll is the pigment that gives plants their green color and plays a crucial role in photosynthesis. It's mainly found in a specific organelle called the chloroplast. Chloroplasts are the sites where sunlight is captured and converted into energy, which is essential for the plant's growth and survival. Understanding where chlorophyll resides helps us appreciate how plants produce their food using sunlight. So remember, the organelle containing chlorophyll is the chloroplast. Answer coming. 5 seconds think. Take a moment to reflect on this question or concept. This pause is designed to give you time to process the information and consider your own perspective. It's a crucial part of understanding how this fits into the bigger picture of our presentation. Remember, engaging actively with these pauses helps deepen your comprehension and retention. So, let's use these few seconds to think carefully before we move forward. Let's take a closer look at the chloroplast. This is the key organelle responsible for photosynthesis in plant cells. It's where light energy is converted into chemical energy, producing glucose and oxygen. You can think of it as the plant's solar panel, capturing sunlight and turning it into something the plant can use. The chloroplast contains a pigment called chlorophyll, which gives plants their green color and plays a crucial role in absorbing light. Understanding chloroplasts helps us grasp how plants produce their own food and sustain life on Earth. This ties into the overall theme of how cellular structures support vital biological processes. So let's talk about protein building blocks. What exactly are they? Well, proteins are made up of smaller units called amino acids. These amino acids are the fundamental components that link together in specific sequences to form proteins. Think of them as the individual Lego bricks that, when assembled in the right order, create complex structures with unique functions. There are 20 different amino acids commonly found in proteins, each with distinct properties. Some are considered essential, meaning our bodies can't produce them, so we need to get them through our diet. Others are non-essential because our bodies can synthesize them ourselves. Understanding these building blocks is crucial because the sequence and types of amino acids determine the protein's structure and function. This connects back to our overall theme of how biological molecules work together to sustain life. Proteins are involved in virtually every process in our bodies, so knowing what they're made of helps us appreciate their complexity and importance. Answer coming soon. Take a moment to think, 5 seconds on the clock. During this time, I want you to consider the key points we've discussed so far. This pause isn't just about waiting, it's about processing and preparing to engage with the upcoming content. Remember, the goal here is to connect what we've learned to the bigger picture of our presentation. So, use these few seconds to reflect on how these concepts fit into the overall theme. This helps reinforce your understanding and sets the stage for the next part of our discussion. Ready? Let's move forward together. Let's take a closer look at amino acids. These are the building blocks of proteins, which are essential for so many functions in our bodies. There are 20 standard amino acids that combine in various ways to form the proteins we need. Some amino acids can be made by our bodies, but others, known as essential amino acids, must come from our diet. Understanding amino acids is fundamental because they influence everything from muscle growth to enzyme activity. Recognizing their role helps us appreciate how our bodies build and repair tissues, and how nutrition directly impacts our health and overall well-being. Let's take a look at this question. What enzyme breaks down hydrogen peroxide, H2O2? This is a really important point because hydrogen peroxide is a byproduct of many metabolic processes, and if it accumulates, it can be harmful to cells. So, the enzyme responsible for breaking down H2O2 is called catalase. Catalase is quite remarkable. It's one of the fastest enzymes in our body. It converts hydrogen peroxide into water and oxygen almost instantly, preventing oxidative damage. Understanding this enzyme helps us see how cells protect themselves from oxidative stress, which ties into the broader theme of cellular defense mechanisms. So, whenever you think about how cells manage reactive oxygen species, remember catalase plays a crucial role. Answer coming. 5 seconds think. All right, take a moment to think about what we've just discussed. This slide is about encouraging a pause, a moment of reflection before we move forward. It's a reminder that sometimes, the best way to understand or solve a problem is to give ourselves a few seconds to process. This simple step can lead to clearer insights and better decisions, which ties into our overall theme of mindful and strategic thinking. So remember, when you're faced with a tough question or a complex situation, don't rush, pause, reflect, and then respond. That's often where the most valuable answers come from. Let's take a closer look at catalase. Catalase is a crucial enzyme in our bodies because it helps break down hydrogen peroxide, which is a harmful byproduct of many metabolic processes. Without it, hydrogen peroxide could accumulate and damage cells. What's really interesting about catalase is that it's incredibly efficient. A single molecule can catalyze the breakdown of millions of hydrogen peroxide molecules every second. This efficiency highlights how vital enzymes are in maintaining cellular health. So, understanding enzymes like catalase gives us insight into how our bodies protect themselves and keep biochemical reactions running smoothly, which ties back to the overall theme of how enzymes facilitate life's essential processes. So let's explore an important question. What is the energy currency of the cell? This is a fundamental concept because cells need energy to perform all their functions. The key player here is a molecule called ATP, or adenosine triphosphate. Think of ATP as the cell's rechargeable battery. It stores energy and is quickly used up when the cell needs to do work. Whenever a cell needs to move things around, produce proteins, or power its processes, it taps into ATP. It's incredibly efficient because ATP can be rapidly regenerated from ADP and phosphate, ensuring a constant supply of energy. Understanding ATP helps us see how cells manage their energy needs seamlessly, supporting everything from basic survival to complex activities. So, in essence, ATP is truly the energy currency that keeps the cell functioning smoothly. Answer coming. Hourglass not done. 5 seconds to think. So, take a moment to consider your response. This slide is a reminder that sometimes, giving yourself a pause to think deeply can lead to better decisions. It's about the value of reflection before action, especially in complex situations. By incorporating this pause, we can improve our problem solving and ensure our responses are well thought out. This concept ties back to the overall theme of mindful decision-making and strategic thinking in our presentation. So, let's remember that a brief pause can make a significant difference in achieving better outcomes. Let's take a closer look at ATP, or adenosine triphosphate. This molecule is often called the energy currency of the cell because it plays such a crucial role in storing and transferring energy. When our cells need energy to perform functions, whether it's muscle contraction, nerve transmission, or biochemical synthesis, ATP is our go-to source. think of ATP as a kind of rechargeable battery. It stores energy in its high-energy phosphate bonds, and when that energy is needed, ATP is broken down into ADP and an inorganic phosphate, releasing energy in the process. This energy then powers various cellular activities. Understanding ATP is fundamental because it ties into the overall theme of how our bodies efficiently manage energy. No matter what function we're focusing on, ATP is always at the core, ensuring that our cells have the energy they need to keep us functioning optimally. Let's take a moment to look at the question, RNA full form. This is a fundamental concept in molecular biology that often comes up. RNA stands for ribonucleic acid, which is crucial for various biological roles. It acts as a messenger that carries genetic information from DNA to the protein-making machinery in cells. Understanding what RNA is and its functions helps us grasp how genetic information is expressed and regulated. So, when you see RNA, remember it refers to ribonucleic acid, an essential molecule in life's processes. Answer coming. 5 seconds think. Now, take a moment to consider what we've just discussed. Think about how these points connect to our broader goals and strategies. This pause is important because it helps us internalize key concepts before moving forward. Remember, understanding these foundational ideas is crucial for the success of our overall plan. So, let's keep that in mind as we continue exploring the next steps together. Let's take a closer look at ribonucleic acid, or RNA. RNA plays a crucial role in the process of translating genetic information from DNA into proteins. Unlike DNA, which is typically double-stranded, RNA is usually single-stranded, allowing it to fold into various shapes that are essential for its functions. One key point to remember is that RNA acts as a messenger, carrying instructions from DNA in the nucleus to the rest of the cell, especially to the ribosomes where proteins are made. It also has different forms, such as messenger RNA, transfer RNA, and ribosomal RNA, each with specific roles in protein synthesis. Understanding RNA helps us see how genetic information is expressed and regulated within cells, which ties back to the overall theme of genetic information, flow, and cellular function. Let's take a moment to clarify the full form of DNA. DNA stands for deoxyribonucleic acid. It's essentially the blueprint of life, carrying all the genetic instructions necessary for the growth, development, and functioning of living organisms. Understanding what DNA stands for helps us appreciate its fundamental role in biology and genetics. So, whenever you hear DNA, remember it's deoxyribonucleic acid, the molecule that contains the information that makes each of us unique. Answer coming. 5 seconds think. Now, take a moment to reflect on what we've discussed so far. This pause is an opportunity to process the key concepts and see how they connect. It's about making sure we really understand the main ideas before moving forward. Remember, this is all part of building a solid foundation for the overall theme of this presentation. So, take these few seconds to think about how these points relate to your own experiences or ideas. This will help you engage more deeply with the material and get the most out of today's session. Deoxyribonucleic acid, or DNA for short, is the molecule that carries the genetic instructions essential for life. It's fascinating because DNA contains the information that determines everything from our physical traits to how our bodies function. Think of it as a biological blueprint that guides the development and operation of all living organisms. The structure of DNA is often described as a double helix, which looks like a twisted ladder, with pairs of bases forming the rungs. These bases, adenine, thymine, cytosine, and guanine, are the key to how genetic information is stored and copied. Understanding DNA is central to grasping how traits are inherited and how genetic variation occurs, which ties into the broader themes of genetics and biology we're exploring in this presentation. So let's talk about the blood oxygen-carrying protein. What exactly is it? Well, It's a crucial component in our blood that helps transport oxygen from our lungs to the tissues throughout our body. The key protein responsible for this function is hemoglobin. Hemoglobin is a complex molecule that contains iron, which is essential for binding oxygen molecules. This process is vital because our cells need oxygen to produce energy and keep us alive and functioning properly. Understanding this protein helps us appreciate how efficiently our circulatory system works. When we breathe in, oxygen enters our lungs and binds to the hemoglobin in our red blood cells. Then, as blood circulates, hemoglobin releases the oxygen to tissues that need it. This exchange is fundamental to our overall health and energy levels. So, in essence, the blood oxygen-carrying protein, hemoglobin, is like the body's delivery truck for oxygen, ensuring every cell gets the fuel it needs. Recognizing its role highlights how interconnected our respiratory and circulatory systems really are, which ties into the broader theme of how our body maintains homeostasis and supports life. Answer coming. 5 seconds think. All right, take a moment to consider your response. This slide is all about the importance of patience and reflection in decision-making. Sometimes, the best choices come after a brief pause to think things through. It reminds us that rushing can lead to mistakes, while taking a moment can lead to better outcomes. This concept ties into our overall theme of strategic thinking and mindful action. So, next time you're faced with a tough decision, remember to pause and reflect. It can make all the difference. Let's now turn our attention to hemoglobin, which is a crucial component in our blood. Hemoglobin is the protein responsible for carrying oxygen from our lungs to the rest of our body. It also plays a key role in returning carbon dioxide from the tissues back to the lungs for exhalation. The structure of hemoglobin is quite fascinating. It's made up of 4 subunits, each containing a heme group that binds oxygen. Because of this, hemoglobin's ability to efficiently pick up and release oxygen is vital for maintaining our overall health. In the context of our presentation, understanding hemoglobin helps us appreciate how our blood functions in oxygen transport and how its levels can indicate different health conditions. So, when we analyze blood tests, hemoglobin levels are one of the first things we look at to assess oxygen-carrying capacity and overall blood health. Let's take a moment to consider the human chromosome number. When we talk about human chromosomes, we're referring to the structures that carry our genetic information. Humans typically have 23 pairs of chromosomes, which totals to 46 individual chromosomes. This number is quite consistent across the human population and is fundamental to our genetic makeup. Understanding this helps us see how our genes are organized and inherited. So, when asked about the human chromosome number, the key point to remember is that it's 46 chromosomes in total. This fundamental fact ties into the broader theme of genetics and heredity, illustrating how our genetic blueprint is stored and passed on. Answer coming. 5 seconds think. Now, take a moment to reflect on what we've discussed so far. This slide is about encouraging you to pause and think deeply before moving forward. It emphasizes the importance of taking that brief pause to process information fully. In the context of our overall theme, this step is crucial for making thoughtful decisions and ensuring clarity. So remember, sometimes the best approach is to slow down just enough to get it right. Let's keep that in mind as we continue. Let's take a closer look at answer 14. This part really emphasizes the importance of thoroughly understanding the key points we've discussed so far. It's about engaging with the material in a way that's conversational and approachable, making sure we highlight what's most important. When we do this, we're connecting these ideas to the broader theme of the presentation, showing how each piece fits into the bigger picture. Remember, the goal is to communicate clearly and confidently, ensuring that our audience walks away with a solid grasp of the core concepts and their relevance. Let's take a closer look at whether cell division actually produces identical cells. This is a fundamental concept because it ties into how organisms grow and repair themselves. When a cell divides, especially during mitosis, the goal is to create 2 new cells that are exact copies of the original. This means they should have the same genetic material and essentially be identical in structure and function. However, it's also worth noting that while the genetic information is copied precisely, there can sometimes be small variations or mutations that occur during DNA replication. But overall, the primary purpose of cell division in this context is to produce genetically identical cells. Understanding this process is crucial because it underpins many biological functions, from tissue growth to healing wounds. It connects directly to the broader theme of how living organisms maintain stability and reproduce themselves. So, in essence, cell division is a key mechanism for creating identical cells, ensuring continuity of genetic information across generations of cells. Answer coming. 5 seconds think. Take a moment to consider your response. Think about the key points we've discussed so far. This pause is a great opportunity to reflect on how this fits into the bigger picture. Remember, the goal is to connect these ideas to our overall theme. So, give yourself a few seconds to gather your thoughts before we move forward. Today, we're going to talk about mitosis, which is a crucial process in cell division. Mitosis is the way our cells replicate to produce two identical daughter cells, ensuring that genetic information is accurately passed on. This process is fundamental for growth, tissue repair, and overall maintenance of our bodies. During mitosis, the cell's DNA is duplicated and evenly divided, which is key to maintaining genetic stability. It's a highly organized series of steps that ensures each new cell has the same genetic material as the original. Understanding mitosis helps us appreciate how our bodies develop and heal, and it also provides insight into how errors in this process can lead to diseases like cancer. So, as we look at this slide, think of mitosis as the cell's way of carefully copying and dividing itself in a controlled manner, vital for life as we know it. So, is cell division producing gametes? That's a great question, and it leads us into an important part of understanding how organisms reproduce. When we talk about gametes, we're referring to the specialized reproductive cells, sperm and eggs in animals, pollen and ovules in plants. These cells are crucial because they carry half the genetic material needed for the next generation. Now, the process that produces these gametes is called meiosis, and it's a specific type of cell division. Unlike mitosis, which creates identical cells for growth and repair, meiosis reduces the chromosome number by half. This reduction is essential because it ensures that when fertilization occurs, the resulting zygote has the correct number of chromosomes. So, in summary, yes, cell division does produce gametes, but it specifically involves meiosis to generate these specialized reproductive cells, linking directly to the broader theme of how genetic information is passed on and maintained across generations. Answer coming. 5 seconds think. All right, take a moment to consider your response. This slide is all about the importance of pausing and reflecting before answering. Sometimes, the most effective communication happens when we give ourselves just a few seconds to gather our thoughts. This practice can lead to clearer, more thoughtful responses and better understanding in any conversation. Remember, it's not just about answering quickly, but about answering wisely. So, whether you're in a meeting, a negotiation, or a casual chat, this simple pause can really make a difference. It ties into our overall theme of mindful communication and strategic thinking. Now, let's move on and see how we can apply this in real-world situations. Let's talk about meiosis. Meiosis is a special type of cell division that's crucial for sexual reproduction. It reduces the chromosome number by half, so when two gametes, like sperm and egg, come together, the resulting zygote has the right amount of genetic material. This process ensures genetic diversity, which is vital for evolution and adaptation. During meiosis, a single cell divides twice, but the DNA replication happens only once, leading to 4 genetically unique daughter cells. This is different from mitosis, which produces identical cells. So, in summary, meiosis plays a key role in creating genetic variation and maintaining the stability of an organism's chromosome number across generations. Let's move on to the study of tissues. So, why is understanding tissues important? Well, tissues are the building blocks of organs and systems, giving them structure and function. In this slide, we're focusing on question 17, which asks about the key points related to tissue study. First, it's essential to recognize that tissues are groups of similar cells working together to perform specific tasks. There are four main types of tissues: epithelial, connective, muscle, and nervous tissue. Each of these has unique characteristics and roles within the body. For example, epithelial tissue covers surfaces and lines cavities, providing protection and function in absorption and secretion. Connective tissue supports and connects other tissues and organs, with examples like cartilage, bone, and blood. Muscle tissue is responsible for movement, and nervous tissue transmits signals throughout the body. Understanding these tissue types helps us grasp how organs develop and function, and it is fundamental to studying health, disease, and medical treatments. Throughout this presentation, we've been exploring how the body is organized from cells to tissues to organs. So, studying tissues is not just about identifying them under a microscope. It's about understanding their roles in maintaining overall health and how disruptions can lead to disease. By mastering tissue study, we gain insights into the structure-function relationship that underpins the entire human body. Answer coming. 5 seconds think. All right, take a moment to gather your thoughts. This slide is all about the anticipation of the answer, and how sometimes we need a little pause to reflect. It emphasizes the importance of pausing before responding, which can lead to more thoughtful and effective communication. Remember, giving yourself that extra few seconds can help you process information better and craft a more precise response. This ties into our overall theme of strategic thinking and deliberate decision-making. So let's keep in mind that sometimes the best answers come when we allow ourselves a moment to think. Let's move on to the next topic, histology. When we talk about histology, we're diving into the microscopic structure of tissues, which is essential for understanding how organs function and how diseases can affect them. Histology allows us to see the detailed architecture of cells and tissues, helping us identify normal versus pathological states. By examining tissues at this level, we can better grasp the organization and specialization within different parts of the body. This understanding is fundamental to many aspects of medicine and biology, tying into the overall theme of how structure relates to function. So, in essence, histology provides the microscopic lens through which we can truly appreciate the complexity and beauty of our body's design. So let's move on to an interesting question. What is the largest organ in the human body? This might seem like a simple fact, but it actually highlights how complex our bodies are. The answer is the skin. It might surprise some people because we often think of internal organs like the heart or lungs as being the largest. But in fact, your skin covers your entire body, acting as a protective barrier. It plays a crucial role in regulating temperature, preventing infections, and even allowing us to sense the environment through touch. Recognizing the skin as the largest organ helps us appreciate how vital it is to our overall health and well-being. It ties into our broader theme of understanding the body's systems and how they work together to keep us healthy. Answer coming. 5 seconds think. Now, take a moment to reflect on what we've just covered. Think about how these concepts connect to the bigger picture we've been discussing throughout the presentation. It's important to consider the key points and how they influence your understanding of the overall topic. So, give yourself a few seconds to process this information before we move forward. Let's talk about the skin. The skin is a fascinating and vital organ, serving as our body's first line of defense. It acts as a barrier, protecting us from environmental hazards like bacteria, chemicals, and physical injuries. The skin also plays a crucial role in regulating body temperature through sweating and blood flow. Additionally, it helps us sense the world around us. Touch, pressure, pain, and temperature are all detected through our skin. Understanding the skin's functions is essential because it ties into the overall health and resilience of the body. It reminds us that taking care of our skin isn't just about appearance, it's about maintaining our overall well-being. So let's talk about where photosynthesis takes place in the cell. This process is vital because it's how plants make their own food, converting sunlight into energy. The key cell part involved here is the chloroplast. Chloroplasts are specialized organelles that capture sunlight and contain the green pigment chlorophyll. This pigment is what gives plants their green color and is essential for absorbing light energy. During photosynthesis, the chloroplasts work to transform carbon dioxide and water into glucose and oxygen. Understanding that photosynthesis occurs in these chloroplasts helps us see how plants are perfectly designed to harness sunlight and sustain life on Earth. Answer coming. 5 seconds think. All right, take a moment to gather your thoughts. In this slide, we're emphasizing the importance of reflection before moving forward. It's about pausing to consider the key points we've just discussed. This step helps to reinforce understanding and ensures we're on the same page. Remember, in the bigger picture, taking a moment to think critically enhances decision-making. So, as we wrap up this section, keep in mind that thoughtful reflection is a vital part of the overall process we've been exploring. Let's take a closer look at the chloroplast. This organelle is essential for photosynthesis, the process that allows plants to convert sunlight into energy. Chloroplasts contain a pigment called chlorophyll, which gives plants their green color and is vital for capturing light energy. Inside the chloroplast, the light-dependent reactions occur, producing energy-rich molecules like ATP and NADPH. These molecules are then used in the light-independent reactions, or the Calvin cycle, to synthesize glucose. Essentially, chloroplasts are the powerhouses of plant cells when it comes to energy production, and they play a crucial role in sustaining life on Earth by producing oxygen and food through photosynthesis. This highlights how integral they are to both plant biology and the larger ecosystem. Let's take a closer look at the process of glucose breakdown. When our body needs energy, glucose is the primary fuel it taps into. The process begins with glycolysis, which occurs in the cytoplasm of the cell. During glycolysis, glucose molecules are broken down into smaller units called pyruvate, generating a small amount of energy in the form of ATP and NADH. This is a crucial step because it prepares the glucose for further energy extraction, especially when oxygen is available. If oxygen is present, pyruvate enters the mitochondria and proceeds through the citric acid cycle, also known as the Krebs cycle. Here, it's further broken down, releasing more energy-rich molecules. Finally, these molecules feed into the electron transport chain, where the majority of ATP is produced. This entire process efficiently converts glucose into usable energy for our cells, supporting everything from muscle movement to brain function. Understanding these steps is key to grasping how our bodies sustain activity and maintain homeostasis. Answer coming. 5 seconds think. Now, take a moment to reflect on what we've just discussed. This slide is all about giving you a little pause to process the key points. It's an opportunity to think critically and connect the dots. Remember, in any project or decision, taking that moment to pause can really enhance your understanding. So, as we move forward, keep in mind how these concepts link back to the overall theme of strategic thinking and effective decision-making. Let's continue and see how we can apply these ideas in real-world scenarios. Glycolysis is a fundamental metabolic pathway that we often start with when discussing how our bodies generate energy. It's essentially the process of breaking down glucose, which is our primary source of fuel, into smaller molecules. During this process, a series of 10 enzymatic reactions occur, ultimately producing pyruvate. This is crucial because pyruvate can then enter other pathways to produce more energy or be used in other cellular functions. One of the key points to remember is that glycolysis occurs in the cytoplasm of the cell, making it accessible for many cell types. It's also an anaerobic process, meaning it doesn't require oxygen, which is vital during short bursts of activity when oxygen might be limited. Another important aspect is that glycolysis produces a net gain of 2 ATP molecules per glucose molecule, which provides immediate energy for cellular activities. Additionally, it generates NADH, a carrier molecule that can be used later in other energy-producing processes. Overall, glycolysis is central to cellular metabolism because it links carbohydrate intake to energy production and supplies intermediates for other metabolic pathways. It's a perfect example of how our bodies efficiently convert food into usable energy, supporting all the functions we rely on every day. So let's talk about which vitamin we get from sunlight. This is a great example of how our body naturally interacts with the environment to support our health. When our skin is exposed to sunlight, specifically ultraviolet B rays, it triggers a process that produces vitamin D. This vitamin is crucial because it helps us absorb calcium, which is essential for strong bones and overall skeletal health. It's interesting to note that vitamin D is often called the sunshine vitamin for this reason. Our body actually synthesizes it on its own, which highlights how important regular, safe sun exposure can be. Of course, we need to balance this because too much sun can have negative effects, but a little bit of sunlight can go a long way in maintaining adequate vitamin D levels. Connecting this to the overall theme, understanding the role of sunlight in vitamin D production underscores how our environment influences our health in subtle but meaningful ways. It also reminds us of the importance of outdoor activity and sunlight exposure, especially in regions where dietary intake might be insufficient. Answer coming. 5 seconds think. Take a moment to consider your response. This pause encourages reflection, helping us internalize the key concepts. It's important to connect this thinking to the overall theme of the presentation, which is about making informed decisions and understanding the bigger picture. By doing so, we reinforce how critical thoughtful analysis is in achieving our goals. So, let's use this time to prepare ourselves for the insights ahead. Let's talk about vitamin D. This vitamin plays a crucial role in our overall health, especially in bone health and immune function. When we think about vitamin D, it's important to remember that our bodies can produce it naturally when our skin is exposed to sunlight. That's why outdoor activity and sensible sun exposure are so beneficial. Vitamin D helps our bodies absorb calcium, which is essential for maintaining strong bones and preventing conditions like osteoporosis. It also supports immune health, helping us fend off illnesses. Sometimes, despite adequate sun exposure, people may still need supplements, especially in regions with limited sunlight during certain seasons. Overall, maintaining optimal vitamin D levels is a key piece of the puzzle when it comes to supporting our health and preventing deficiencies. It ties directly into the broader theme of how nutrition and lifestyle choices influence our well-being. Let's talk about the human heart chambers. So, what exactly are these chambers? Well, the heart is divided into 4 main sections that work together to pump blood effectively throughout the body. The 2 upper chambers are called the atria. There's the right atrium and the left atrium. These chambers receive blood coming into the heart. The 2 lower chambers are called the ventricles, again, a right ventricle and a left ventricle. These are responsible for pumping blood out of the heart to the lungs and the rest of the body. Understanding these chambers is key because each plays a vital role in the circulatory process. The right atrium receives deoxygenated blood from the body and passes it to the right ventricle, which then pumps it to the lungs for oxygen. Meanwhile, the left atrium gets oxygenated blood from the lungs and sends it to the left ventricle, which then pumps it out to supply the entire body. This division into chambers ensures that blood flow is efficient and properly directed, crucial for maintaining healthy circulation. So, in summary, the heart's 4 chambers work in a coordinated way to keep blood moving smoothly, supporting all the functions we've discussed so far. Answer coming. 5 seconds think. All right, take a moment to gather your thoughts, because what we're about to explore is quite important. This slide is a reminder that sometimes the best insights come after a brief pause for reflection. It encourages us to think deeply about the key points we've discussed so far and how they connect to the bigger picture. By pausing and contemplating, we can better understand the core concepts and see how they fit into our overall strategy. Remember, thoughtful consideration often leads to more informed decisions, especially in complex situations. So, let's use this moment not just as a break, but as an opportunity to solidify our understanding before moving forward. Let's take a closer look at question 22 now. This question is a great example of how we can apply the concepts we've discussed so far. It highlights 4 key points that are crucial for understanding the overall theme of this presentation. As we go through each point, I want you to think about how they connect to the bigger picture and the practical implications. Remember, the goal here is to deepen your understanding and see how these pieces fit together to form a comprehensive approach. So, let's dive into each of these 4 answers and explore their significance in our broader discussion. So, what are genetic units called? Well, these fundamental units are known as genes. Genes are segments of DNA that carry the instructions for building and maintaining our bodies. They determine everything from our physical features to certain aspects of our health. Understanding genes is crucial because they are the basic units of heredity, passing traits from one generation to the next. This concept ties into the larger theme of how genetic information influences life, evolution, and even medical science. Recognizing these units helps us appreciate the complexity and beauty of biological inheritance. Answer coming. 5 seconds think. Now, take a moment to reflect on what we've discussed so far. This slide is about emphasizing the importance of patience and careful consideration. When we're faced with complex decisions or challenging problems, giving ourselves a little extra time to think can make a big difference. It helps us avoid rushing into mistakes and allows us to see the situation from different angles. Remember, the key is not just to answer quickly, but to provide thoughtful, well-informed responses. This ties into the overall theme of strategic thinking and deliberate action. So next time you're unsure, just pause, take a few seconds, and really consider your options. It's a simple step that can lead to better outcomes and more effective problem solving. Let's talk about genes. Genes are essentially the instructions within our DNA that determine many aspects of who we are. They carry the information needed to build and maintain our bodies. When we answer question 23, we're delving into the significance of these genetic instructions and how they influence traits, health, and even susceptibility to certain diseases. Understanding genes helps us see how biological information is passed from one generation to the next, shaping individual differences. This concept ties into the broader theme of genetics and heredity, highlighting the fundamental role genes play in biology and medicine. So, by examining genes closely, we gain insights into the very blueprint of life itself. Let's take a closer look at plant water transport tissue. This is a fundamental aspect of how plants function, especially when it comes to moving water from the roots up to the leaves. Water transport tissue in plants primarily consists of xylem vessels, which are specialized for conducting water efficiently. These vessels form a continuous system that stretches from the roots through the stem and into the leaves. Understanding this system is key because water movement is essential for photosynthesis, nutrient transport, and overall plant health. The process relies on a combination of root pressure, capillary action, and the transpiration pull from the leaves. So, when we ask about plant water transport tissue, we're really talking about how plants have evolved these specialized structures, like xylem, to meet their water needs. This system is a great example of how plants have adapted their anatomy to survive and thrive in diverse environments. It ties directly into our broader theme of plant physiology and how structural features support vital functions. Answer coming. 5 seconds think. All right, take a moment to consider your thoughts. This pause is designed to give you a chance to reflect on what we've just discussed. It's important because it helps solidify your understanding before we move forward. Remember, the key points we've covered so far are central to grasping the overall theme of this presentation. So, use this time wisely to connect the dots and think about how these ideas fit into the bigger picture. When you're ready, we'll continue exploring the next steps together. Let's talk about xylem. Xylem is a type of tissue in plants that plays a crucial role in transporting water and minerals from the roots up through the stem and into the leaves. It's essentially the plant's plumbing system, ensuring that all parts of the plant receive the water they need to carry out their functions. The structure of xylem includes specialized cells like tracheids and vessel elements, which are designed to facilitate efficient water conduction. Understanding how xylem works helps us appreciate the incredible adaptations plants have evolved to survive and thrive in various environments. This ties into the broader theme of how plants manage resource transport and maintain their structural integrity. So let's talk about the tissue responsible for transporting food in plants. Now, this is a key part of how plants distribute nutrients from the leaves, where photosynthesis happens, to other parts of the plant that need energy and building blocks. The tissue we're focusing on is called the phloem. Phloem is specialized for the movement of organic substances, mainly sugars like sucrose, which are produced in the leaves. Think of it as the plant's delivery system for food. Unlike xylem, which carries water and minerals upward, phloem transports these sugars downward or to growing parts of the plant. The process involved here is called translocation. It's quite fascinating because it involves active loading of sugars into the phloem, creating a pressure difference that pushes the food along the tissue. This movement is essential for the overall health and growth of the plant, ensuring all parts get the nutrients they need. Understanding this transport system really highlights how plants are so well adapted to efficiently distribute resources, which ties into the bigger picture of how they sustain themselves and grow. It's a perfect example of the complexity behind what might seem like simple processes at first glance. Answer coming soon, so hang tight. Take a moment to think about what we've discussed so far. You've got 5 seconds to really process the key ideas. This pause is important because it helps you connect the dots and prepare for the next part. Remember, the overall theme is about understanding how these concepts fit into the bigger picture. So, use this time to reflect on how everything you've learned so far contributes to the larger goal. And when you're ready, we'll move forward together. Let's take a closer look at the phloem. The phloem is a crucial part of the plant's vascular system. Its main role is to transport sugars and other organic nutrients produced in the leaves to different parts of the plant. This process is known as translocation. Unlike xylem, which mainly moves water and minerals upward, the phloem can move substances in both directions, depending on the plant's needs. This dual transport capability is vital for supporting growth, storage, and energy distribution throughout the plant. Understanding the phloem helps us appreciate how plants efficiently distribute the products of photosynthesis, ensuring their overall health and productivity. So, have you ever wondered what gas plants actually take in during photosynthesis? Well, this is a really interesting part of how plants make their food. The key gas involved here is carbon dioxide. Plants absorb carbon dioxide from the air through tiny openings on their leaves called stomata. This gas is essential because it reacts with water inside the plant to produce glucose, which is their main source of energy. Remember, photosynthesis is all about converting light energy into chemical energy, and carbon dioxide is a vital ingredient in that process. So, in summary, the gas taken in by plants for photosynthesis is primarily carbon dioxide, and this process is central to the overall theme of how plants sustain themselves and contribute to the Earth's ecosystem. Answer coming. 5 seconds think. Take a moment to reflect on what we've discussed so far. This slide is all about encouraging you to pause and consider the key points carefully. In the context of our overall theme, it's crucial to understand that thoughtful analysis leads to better decision-making. So, I invite you to really think about how these concepts connect and what implications they might have for your own work or projects. Remember, sometimes taking a few seconds to process information can make all the difference in applying it effectively. Let's take a moment to focus on carbon dioxide. As you know, it's a significant greenhouse gas that plays a key role in our climate system. Carbon dioxide is produced through various human activities, especially burning fossil fuels like coal, oil, and natural gas. This process releases large amounts of CO2 into the atmosphere, contributing to global warming. It's important to understand that while CO2 is naturally present in our atmosphere and necessary for processes like photosynthesis, the excessive amounts caused by human activity are what drive climate change. Recognizing the role of carbon dioxide helps us see why reducing emissions is so critical for protecting our environment and ensuring a sustainable future. Let's explore how the unit of enzyme activity is affected by pH and temperature. This is a crucial aspect because enzymes are highly sensitive to their environment. When we talk about enzyme activity, we're referring to how effectively an enzyme can catalyze a reaction. Now, both pH and temperature can influence this activity significantly. For example, each enzyme has an optimal pH at which it functions best. Deviating from this pH can cause the enzyme's structure to change, reducing its efficiency. Think of it like a lock and key. If the shape of the lock or key changes, the fit isn't perfect anymore. Similarly, temperature affects enzyme activity because increasing temperature generally increases reaction rates up to a point, but too much heat can denature the enzyme, messing up its structure. So, the key takeaway here is that enzymes have specific pH and temperature ranges where they perform optimally. Outside of these ranges, their activity drops, which is critical to consider in both biological systems and industrial applications. This understanding helps us appreciate how finely tuned enzymes are to their environments and why maintaining proper conditions is essential for their function. Answer coming. 5 seconds think. All right, take a moment to gather your thoughts. This slide is about the importance of pausing and reflecting before we move forward. In many situations, especially when dealing with complex decisions or challenges, a brief pause can be incredibly valuable. It gives us time to consider all the key points, weigh our options, and ensure we're aligned with our overall goals. By intentionally taking a moment to think, we enhance our clarity and avoid rushing into actions that might not serve us well. So remember, sometimes the best move is to pause, reflect, and then proceed with confidence. This concept ties back to the overall theme of thoughtful decision-making and strategic planning throughout the presentation. Let's take a closer look at enzyme activity. Enzymes are fascinating because they act as biological catalysts, speeding up chemical reactions in our bodies. Their activity can be influenced by various factors such as temperature, pH, substrate concentration, and the presence of inhibitors or activators. When conditions are optimal, enzymes work most efficiently, allowing vital processes like digestion, energy production, and cell signaling to occur smoothly. Understanding enzyme activity is key to grasping how biological systems maintain homeostasis and respond to changing conditions. So, in essence, enzyme activity is all about how effectively these proteins facilitate life's essential reactions under different circumstances. Let's talk about protein separation techniques based on size. When we want to separate proteins, one effective method is size-based separation, which is often achieved through gel electrophoresis. This technique allows us to distinguish proteins by their molecular weight, with smaller proteins migrating faster through the gel matrix, while larger ones move more slowly. It's a crucial tool in biochemistry because it helps us analyze protein purity, identify proteins, and estimate their molecular sizes. Connecting this to the overall theme, understanding how proteins separate by size is fundamental to many experiments, from diagnosing diseases to studying protein structures. So, grasping this concept helps us better interpret experimental results and advances our insights into protein function. Answer coming. 5 seconds think. Now, take a moment to reflect. There's a reason we're asking you to pause here. This is a chance to really consider what's been discussed so far and how it connects to the bigger picture. think about the key points, the concepts that stand out, and how they all tie together. It's about engaging your mind and making those connections. This quiet moment reinforces the importance of thoughtful analysis in our overall approach. Remember, the goal is not just to absorb information, but to understand how it applies in real-world scenarios. So, take this time to process and prepare for the next part of our presentation. Today, let's take a closer look at gel filtration chromatography, which is also known as size exclusion chromatography. This technique is primarily used to separate molecules based on their size. The process involves a column filled with porous beads, and as the mixture passes through, smaller molecules enter the pores and take longer to travel, while larger molecules are excluded from the pores and exit the column more quickly. This allows us to effectively sort our molecules by size. One of the key advantages of gel filtration chromatography is that it's gentle and maintains the native structure of proteins, making it ideal for studying their functional forms. It's also highly useful for desalting and buffer exchange, which are common steps in preparing samples for further analysis. Connecting this to our overall theme, gel filtration chromatography is a vital tool in protein purification and analysis, helping us understand the size distribution and quaternary structure of proteins in complex mixtures. It's a fundamental technique that complements other separation methods, providing a clear window into the molecular world. So let's talk about protein separation by molecular weight. This is a fundamental technique in biochemistry and molecular biology. Essentially, we're interested in how proteins can be separated based on their size, specifically their molecular weight. This allows us to analyze complex mixtures and identify individual proteins. The most common method for this is gel electrophoresis, specifically SDS-phage. In this process, proteins are first denatured and coated with SDS, which gives them a uniform negative charge proportional to their length. This means when we apply an electric field, proteins will migrate through the gel primarily based on their size. Smaller proteins move faster and farther, larger ones move more slowly. By running a molecular weight marker alongside our samples, we can compare the migration distances and accurately estimate the size of our proteins. This technique is incredibly useful because it provides a quick visual way to assess protein purity, size, and even to compare expression levels. Overall, understanding how proteins separate by molecular weight helps us interpret experimental data, troubleshoot purification protocols, and better understand protein structure and function in the context of the whole system. Answer coming. 5 seconds think. All right, take a moment to gather your thoughts. This slide is all about the importance of patience and reflection in our decision-making process. When we're faced with complex problems, giving ourselves a few seconds to think can make a huge difference. It helps us avoid impulsive choices and encourages a more thoughtful approach. Remember, quick reactions are not always the best. Sometimes, pausing and considering the options leads to better outcomes. This idea ties into our overall theme of strategic thinking and deliberate action. So, next time you're in a situation that requires a decision, try to incorporate that brief pause. It might just be the key to making smarter, more effective choices. Let's take a closer look at SDS-phage, which stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. This technique is fundamental for analyzing proteins. It allows us to separate proteins based on their molecular weight by applying an electric current through a gel matrix. The SDS detergent denatures proteins and gives them a uniform negative charge, so they migrate through the gel primarily according to size. Smaller proteins travel faster and move farther than larger ones. This method is incredibly useful for identifying, quantifying, and assessing the purity of protein samples. By comparing the migration of unknown proteins to known standards, we can determine their approximate molecular weights. Overall, SDS-phage is a cornerstone technique in biochemistry and molecular biology labs for protein analysis. All right, let's talk about antibody detection techniques. So, when we're trying to identify if someone has been exposed to a specific pathogen, we often look for antibodies in their blood. These techniques are crucial because they help us understand immune responses and determine past infections. One common method is the enzyme-linked immunosorbent assay, or ELISA, which is highly sensitive and specific. It works by binding antibodies to antigens fixed on a surface, then using enzyme-linked secondary antibodies to produce a measurable signal. Another technique is the Rapid Diagnostic Test, or RDT, which is quick and easy to use, often giving results within minutes. These are especially useful in field settings or resource-limited environments. Then there's the Western blot, which is more detailed and used for confirmatory testing. It separates proteins by size and detects specific antibodies, giving us a clearer picture of the immune response. Overall, antibody detection techniques are vital tools in our diagnostic arsenal, helping us track infections, monitor vaccine responses, and improve public health interventions. They're a key part of understanding the immune landscape in any outbreak or disease study. Answer coming soon. Take a moment to think, 5 seconds. This pause helps us gather our thoughts and prepare for the key points we're about to discuss. It's a great way to ensure we're engaging deeply with the material. As we wait, remember that understanding these concepts is essential for connecting the dots in the bigger picture of our presentation. So, let's use this time to reflect, and when you're ready, we'll dive into the details together. Let's take a moment to look at the Western blot, which is a fundamental technique used in molecular biology. Essentially, it allows us to detect specific proteins within a complex mixture. The process involves separating proteins by size using gel electrophoresis, then transferring them onto a membrane. From there, we use antibodies that specifically bind to our target proteins, and through a detection system, we can visualize these proteins as distinct bands. Western blotting is incredibly powerful because it not only confirms the presence of a protein, but also gives us an idea of its relative abundance. This technique is central to understanding protein expression, modifications, and interactions, making it a cornerstone in many research studies. So, when we see a Western blot, we're essentially looking at a protein fingerprint, revealing valuable information about the molecular makeup of our samples. Let's take a moment to reflect on the study of microorganisms. Why is this area so important? Well, microorganisms are all around us, and they play crucial roles in health, disease, and even the environment. Understanding them helps us develop better treatments, improve sanitation, and advance biotechnology. When we study microorganisms, we're essentially unlocking the secrets of some of the tiniest yet most impactful organisms on Earth. This knowledge connects directly to broader themes like disease prevention, innovation in medicine, and ecological balance. So, in essence, studying microorganisms is fundamental to many aspects of science and healthcare. Answer coming. Hourglass not done. 5 seconds to think. Now, let's take a moment to reflect on what this means. The key point here is that sometimes, the best answers or solutions come after a bit of pause and consideration. It's not always about rushing to respond, but rather about giving ourselves the time to process information thoroughly. This approach can lead to more thoughtful, effective decisions, which ties into the overall theme of strategic thinking and mindful decision-making. So remember, sometimes the most valuable insights come from that pause, those few seconds we take to think deeply. Let's take a moment to discuss microbiology, which is a fundamental aspect of understanding infectious diseases and how our bodies respond to them. Microbiology involves studying tiny organisms like bacteria, viruses, fungi, and parasites that can cause illness. Recognizing these microorganisms helps us develop better prevention and treatment strategies. It's also crucial for understanding how infections spread and how we can control outbreaks. Microbiology ties directly into the overall theme of infection control, emphasizing the importance of hygiene, sterilization, and vaccination. By understanding the microbial world, we can better protect ourselves and improve public health outcomes. All right, let's talk about the largest cell organelle. So, what do you think it might be? Well, the answer is the nucleus. The nucleus is often considered the control center of the cell because it houses the genetic material, DNA. This DNA contains all the instructions the cell needs to function and reproduce. It's quite large compared to other organelles, which is why it stands out. Understanding the nucleus helps us grasp how cells coordinate activities and pass on traits, tying into the broader theme of cellular organization and function. So, whenever you're thinking about cell structure, remember the nucleus is the biggest and one of the most vital parts. Answer coming. 5 seconds think. All right, take a moment to really consider the question or the topic at hand. This pause is designed to give you time to reflect, analyze, and prepare a thoughtful response. It emphasizes the importance of deliberate thinking in problem-solving and decision-making processes. Remember, pausing like this can help us approach complex issues more clearly and confidently. So, let's use these few seconds to focus and get ready to engage meaningfully with the content. Let's take a closer look at the nucleus. This is essentially the control center of the cell. It contains most of the cell's genetic material, the DNA, which carries the instructions for how the cell functions and how it reproduces. The nucleus is vital because it regulates gene expression and maintains the integrity of these genetic instructions. Think of it as the command hub that directs all the activity within the cell. Understanding the nucleus helps us see how cells grow, divide, and respond to their environment, which ties back to the overall theme of cellular function and health. Let's explore the question, what is the outer rigid layer of a plant cell? This layer is crucial because it provides structural support and protection. In plant cells, this outer layer is known as the cell wall. It's made primarily of cellulose, a strong carbohydrate that gives the cell its shape and helps it withstand pressure. This cell wall is different from the membranes we see in animal cells because it's rigid and sturdy. Its presence is essential for maintaining the plant's overall integrity, especially as it grows and faces environmental stresses. Recognizing this layer helps us understand how plants maintain their structure and how they differ fundamentally from animal cells. Answer coming. Hourglass not done. 5 seconds to think. Now, as we consider this slide, I want to emphasize that sometimes the most valuable insights come when we pause and reflect. This moment of waiting encourages us to process what we've learned so far and prepare for the next steps. It also highlights the importance of patience and deliberate thinking in our overall approach. Remember, taking time to think critically is a key part of making informed decisions and driving meaningful progress. So, let's keep this in mind as we move forward in the presentation. Let's take a closer look at the cell wall. The cell wall is a vital structure that surrounds certain cells, providing them with support and protection. It acts like a sturdy shield, maintaining the cell's shape and preventing it from bursting due to internal pressure. In plants, the cell wall is primarily made of cellulose, which gives it strength and rigidity. Fungi have cell walls made of chitin, while bacteria typically have cell walls composed of peptidoglycan. Understanding the composition and functions of the cell wall helps us appreciate how different organisms maintain their structural integrity and adapt to their environments. This structure is fundamental to the overall health and function of these cells, tying into the bigger picture of cellular biology and organismal survival. So, is the human brain the largest part of the body? Well, actually, no. The brain is incredibly important, but in terms of size, the largest part of the human body is the skin, which covers and protects everything inside. When we look at internal organs, the liver is the heaviest, but in terms of overall size, the brain might seem large because of its complexity, but it's not the biggest. Understanding the relative sizes of body parts helps us appreciate how our body functions as a whole and how different organs contribute to our health and daily life. This connection really ties into the overall theme of how our body systems work together seamlessly, each with its own role, no matter the size. Answer coming. 5 seconds think. Now, let's take a moment to really consider this question. Think about what we've discussed so far and how it connects to the bigger picture. This pause is important because it helps us process the key points more deeply. Remember, the goal is to understand not just the facts, but how they fit together within the overall theme. So, take these few seconds to reflect, and when you're ready, we'll move forward with clarity. Let's take a closer look at the cerebrum. This is the largest part of the brain and is responsible for many of the higher brain functions. When we talk about the cerebrum, we're referring to the area that controls things like thinking, reasoning, problem-solving, and voluntary movement. It's divided into two hemispheres, the left and the right, each specialized for different functions, but working together seamlessly. The surface of the cerebrum is called the cerebral cortex, which is made up of gray matter and is involved in complex cognitive processes. This structure is essential because it integrates sensory information, coordinates muscle activity, and plays a key role in our consciousness and personality. Understanding the cerebrum helps us appreciate how our brain supports the wide range of human abilities that are central to our overall function and experience. Let's dive into the topic of immune defense proteins. So, what are these proteins exactly? Well, they are special molecules that our bodies produce to help fight off infections and protect us from disease. They play a crucial role in our immune response, acting as the body's natural defense system. These proteins include a variety of types, such as antibodies, which recognize and neutralize pathogens, and complement proteins that assist in destroying invaders. What makes them particularly interesting is how they work together to identify, attack, and eliminate foreign organisms. Understanding these proteins is key to appreciating how our immune system maintains health and combats illness. Throughout this presentation, we see how these defense proteins are central to the overall immune response and how they contribute to our body's ability to stay resilient against infections. Answer coming. 5 seconds think. Now, take a moment to really consider what we've just discussed. This slide is about encouraging you to pause and reflect before moving forward. It's a reminder that sometimes, the best insights come when we give ourselves a little extra time to think. By doing so, we can deepen our understanding and make more informed decisions. Remember, in the context of this presentation, thoughtful reflection is key to effective action. So, I invite you to hold that question or idea in your mind for a few seconds before you continue. This small pause can make a big difference in how we process information and apply it. Let's talk about antibodies. These are specialized proteins produced by our immune system to recognize and neutralize foreign invaders like bacteria and viruses. Think of them as our body's targeted defense agents. When an invader enters, antibodies identify specific parts of it, called antigens, and mark it for destruction. This process is crucial for our immune response and helps prevent infections from spreading. Understanding antibodies is essential because they play a key role not only in natural immunity, but also in medical applications like vaccines and antibody-based therapies. They truly are a cornerstone of our immune defense system. So let's take a closer look at blood clotting and the role of cell fragments. When we think about blood clotting or coagulation, it's a highly coordinated process that involves more than just the blood cells themselves. One key player are the cell fragments, which are actually derived from larger cells called megakaryocytes. These fragments are known as platelets, and they're essential for stopping bleeding. Platelets are not whole cells. Rather, they're small, disc-shaped fragments that circulate in our blood. When a blood vessel is injured, these platelets quickly adhere to the damaged site, become activated, and release chemicals that amplify the clotting process. They also aggregate, or stick together, to form a temporary plug that helps seal the injury. This process is crucial because it prevents excessive blood loss and initiates the healing process. So, in essence, these cell fragments, platelets, are vital components of our blood clotting system, acting as the first responders to vascular injury. They connect directly to the overall theme of how our body maintains homeostasis and protects us from bleeding out. Answer coming. 5 seconds think. Take a moment to consider this question carefully. Think about the key points we've discussed so far. Reflect on how they connect to the overall theme of the presentation. This pause is an opportunity to deepen your understanding. Remember, the goal is to grasp not just the facts, but the bigger picture. So, take these few seconds to process and prepare for the next part. Ready? Let's move forward. Let's take a closer look at platelets. These tiny cell fragments are crucial for blood clotting and wound healing. Although they're much smaller than red or white blood cells, their role is incredibly vital. When a blood vessel is injured, platelets quickly gather at the site, adhere to the damaged area, and form a plug to prevent excessive bleeding. They also release chemicals that help activate other platelets and promote the clotting process. Understanding how platelets function helps us appreciate their importance in maintaining our body's hemostasis. So, in the broader context of blood components, platelets are essential even though they're few in number. Their rapid response and ability to facilitate healing make them a key element in our circulatory system's defense mechanism. Let's talk about sugar in DNA. You might be wondering, is there actually sugar involved in DNA? Well, the answer is yes. The key component of DNA's structure includes a sugar called deoxyribose. This sugar forms the backbone of the DNA molecule and plays a crucial role in its stability and function. It connects with phosphate groups and nitrogenous bases to create the famous double helix structure. So, when we think about the building blocks of DNA, the sugar is an essential part that helps give DNA its shape and integrity. Understanding this helps us see how DNA is constructed and how it carries genetic information. Answer coming. 5 seconds think. Take a moment to gather your thoughts, and while you do, keep in mind that this slide is about the importance of patience and reflection in our process. It's often during these pauses that we gain clarity and insight. Remember, the key points here are about allowing ourselves the time to think deeply before making decisions. This connects to our overall theme of strategic planning and deliberate action. So, let's embrace these moments of silence as valuable opportunities for growth and better choices. Deoxyribose, that's the key sugar component of DNA. It's a 5-carbon sugar, which means it has 5 carbon atoms in its structure. This sugar is crucial because it forms the backbone of the DNA molecule, providing stability and support for the genetic information. The name deoxyribose actually hints at its structure. It's similar to ribose, but with one less oxygen atom, which makes a big difference in how DNA functions compared to RNA. Understanding deoxyribose helps us appreciate how the structure of DNA is built and why it's so well suited to store and transmit genetic information across generations. So, in essence, deoxyribose is more than just a sugar. It's a fundamental building block of life's blueprint. Let's take a closer look at the question, sugar in RNA? This is an interesting point because, unlike DNA, RNA contains a different type of sugar. Specifically, RNA has ribose, which differs from deoxyribose in DNA by having a hydroxyl group on the 2-carbon. This small structural difference has significant implications for RNA's stability and function. The presence of this hydroxyl group makes RNA more reactive and less stable under certain conditions, which is why it is typically more transient in cells. So, when we talk about sugar in RNA, we're referring to ribose, and understanding this helps us appreciate why RNA behaves differently from DNA in biological systems. This detail ties into the overall theme of how molecular structure influences function in genetic material. Answer coming. 5 seconds think. All right, take a moment to gather your thoughts. This slide is about the importance of patience and reflection before jumping to conclusions. In many situations, giving ourselves a few seconds to pause can lead to better decisions. It's about slowing down enough to consider all the key points and possible outcomes. This connects to our overall theme of mindful decision-making and strategic thinking. Remember, a well-thought-out response often beats a quick one. So take that extra moment. You'll thank yourself later. Let's take a closer look at ribose. Ribose is a crucial sugar in biochemistry, especially because it's a key component of RNA. It's a 5-carbon sugar, which means it has 5 carbon atoms in its structure, making it a pentose sugar. This sugar provides the backbone for nucleotides, the building blocks of genetic material. Without ribose, RNA couldn't function properly, and neither could many other vital biological processes. So, understanding ribose helps us see how genetic information is stored and transferred at the molecular level. It's a great example of how a simple sugar plays such an essential role in life's complexity. All right, let's dive into this question. What is the enzyme that synthesizes DNA? Well, the key enzyme responsible for building new DNA strands during replication is called DNA polymerase. This enzyme reads the existing DNA strand and adds complementary nucleotides to create a new, identical copy. It plays a crucial role in ensuring the accuracy and efficiency of DNA replication, which is fundamental for cell division and genetic inheritance. Remember, without DNA polymerase, the process of copying genetic material would be impossible. So, understanding this enzyme helps us grasp the core mechanisms that keep our genetic information intact and faithfully passed on. Answer coming. 5 seconds think. Now, let's take a moment to reflect on what we've just discussed. This slide is all about the importance of patience and careful consideration before jumping to conclusions. Sometimes, the best answers come when we give ourselves a little time to think. It highlights that in problem-solving or decision-making, pausing can lead to more thoughtful and effective outcomes. Remember, rushing often leads to overlooked details or overlooked opportunities. So, as we move forward, keep in mind that a brief pause can make a big difference in the overall success of our strategies. This idea connects directly to the broader theme of deliberate and mindful decision-making throughout this presentation. Let's talk about DNA polymerase, which is a crucial enzyme in DNA replication. This enzyme is responsible for synthesizing a new DNA strand by adding nucleotides one by one, following the template strand. It essentially reads the existing DNA and constructs a complementary copy. What's fascinating is that DNA polymerase also has proofreading abilities, meaning it can detect and correct errors during replication, which helps maintain genetic stability. Understanding how DNA polymerase works is key to grasping the overall process of DNA replication and how genetic information is accurately passed on. It highlights the precision and efficiency of cellular mechanisms that keep our genetic information intact. Let's explore the enzyme responsible for RNA synthesis. When we talk about RNA synthesis, we're referring to the process of creating RNA molecules from a DNA template. The key enzyme involved here is RNA polymerase. This enzyme plays a crucial role because it reads the DNA sequence and constructs a complementary RNA strand. RNA polymerase is fascinating because it doesn't require a primer to start, unlike DNA polymerase. It binds to specific regions of DNA called promoters, which help regulate where transcription begins. Once bound, RNA polymerase unwinds the DNA strands and synthesizes the RNA in the 5 to 3 direction, adding nucleotides complementary to the DNA template strand. Understanding this enzyme is essential because it's central to gene expression, allowing cells to produce proteins based on genetic information. So, in essence, RNA polymerase is the molecular machine that drives the transcription process, connecting our genetic code to the functional molecules that keep cells alive and functioning. Answer coming. 5 seconds think. Now, take a moment to reflect on what we've discussed so far. This slide is all about the importance of patience and deliberate thinking in decision-making. Sometimes, rushing can lead us astray, so pausing for a few seconds allows us to gather our thoughts. By doing so, we ensure our choices align with our overall goals and strategies. Remember, this concept ties into the bigger picture of making smarter, more effective decisions in our work and personal lives. So, let's keep this in mind as we move forward in the presentation. Let's take a closer look at RNA polymerase. This enzyme is essential for the process of transcription, where the genetic information in DNA is converted into RNA. Essentially, RNA polymerase reads the DNA template strand and synthesizes a complementary RNA strand. It's like a molecular machine that unzips the DNA and strings together the RNA nucleotides in the correct sequence. One of the most important points to remember is that RNA polymerase doesn't require a primer to start transcription, unlike DNA replication. It binds specifically to certain regions of the DNA called promoters, which signal the start of a gene. Additionally, RNA polymerase plays a crucial role in gene expression, controlling which genes are turned on or off in a cell. Its activity determines the production of proteins, and ultimately, how the cell functions and responds to its environment. Understanding how RNA polymerase works helps us grasp the fundamental process of gene regulation, which is central to all of biology. So, it's not just an enzyme, it's a key player in the flow of genetic information from DNA to RNA, and ultimately to proteins. So, where does protein synthesis actually take place in the cell? This is a great question because understanding the site of this process is key to grasping how proteins are made. Well, the main site is the ribosome, which can be found either floating freely in the cytoplasm or attached to the endoplasmic reticulum, specifically the rough ER. The reason the rough ER is called rough is because of the ribosomes attached to its surface. These ribosomes are the cellular machinery that reads messenger RNA and assembles amino acids into proteins. So, whether free in the cytoplasm or bound to the ER, ribosomes are the cellular factories where proteins are synthesized. Recognizing this helps us see how cellular organization supports efficient protein production, which is fundamental to cell function and overall organism health. Answer coming soon. Take a moment, 5 seconds to think. This pause is important because it allows us to reflect on the question or topic at hand. When we take that time, we're engaging our minds more deeply, which helps us arrive at better insights. Remember, the key points here are about patience and deliberate thinking. Connecting this to our overall theme, it's a reminder that meaningful decisions and innovations often come from taking that extra moment to consider all angles. So, let's keep that in mind as we move forward in our discussion. Let's take a closer look at the ribosome. This tiny but mighty structure is essential for protein synthesis in all living cells. It acts as the site where messenger RNA, or mRNA, is translated into a sequence of amino acids to form proteins. Think of it as the cell's protein factory. The ribosome reads the instructions carried by mRNA and assembles the corresponding amino acids with incredible precision. Highlighting its importance, without ribosomes, cells wouldn't be able to produce the proteins necessary for growth, repair, and overall function. Understanding ribosomes helps us grasp how genetic information ultimately results in the proteins that keep us alive and functioning. So, who actually discovered the double helix structure? Well, it was James Watson and Francis Crick, who, back in 1953, proposed the famous double helix model of DNA. Their discovery was a huge breakthrough, fundamentally changing our understanding of genetics and heredity. It's really fascinating how they pieced together the clues from X-ray diffraction images, especially those taken by Rosalind Franklin, to build this elegant model. This discovery not only explained how genetic information is stored, but also how it can be replicated accurately. It's a perfect example of scientific collaboration and how different pieces of research come together to unlock life's mysteries. Understanding this structure is central to grasping how genetic information is passed down through generations, linking directly to the larger themes of biology and genetics we've been exploring. Answer coming. 5 seconds think. All right, take a moment to gather your thoughts. This slide is all about the importance of patience and reflection in our decision-making process. Sometimes, the best answers aren't immediate. They come after we take a little time to think. By pausing, we can evaluate our options more thoroughly and avoid impulsive reactions. This concept ties into the overall theme of strategic thinking and careful planning. Remember, Giving yourself that extra moment can lead to better outcomes and more thoughtful choices. So don't rush, use that pause wisely. Let's take a moment to focus on Watson and Crick, the scientists who made a groundbreaking discovery in the field of genetics. They are famously credited with deciphering the structure of DNA, which was a major milestone. Their work revealed the double helix shape, helping us understand how genetic information is stored and replicated. Now, you might wonder why I mentioned answer 42. Well, that's a fun nod to the idea of seeking answers in complex questions, much like how Watson and Crick unraveled the mystery of DNA. Their discovery laid the foundation for modern genetics and has had profound implications across biology and medicine. So, understanding their contribution is key to appreciating how we've come to grasp the blueprint of life itself. So, let's talk about what the cell membrane is mainly made of. This is a fundamental question because the membrane plays such a crucial role in protecting the cell and controlling what enters and exits. The primary component of the cell membrane is a phospholipid bilayer. These phospholipids have a hydrophilic, or water-loving, head and two hydrophobic, or water-fearing, tails. This arrangement creates a semi-permeable membrane that allows certain molecules to pass through while blocking others. In addition to phospholipids, the membrane also contains proteins, which are essential for various functions like transport, signaling, and structural support. Cholesterol is another key component. It helps maintain the membrane's fluidity and stability across different temperatures. So, when we think about the overall structure, the cell membrane is mainly made of phospholipids, but it's a complex, dynamic structure that includes proteins and cholesterol as well. These components work together to ensure the membrane functions properly, supporting the idea that the membrane is not just a barrier, but an active, responsive part of the cell. Answer coming soon. Let's take a moment to think about this. Hourglass not done, 5 seconds to reflect. Now, as we consider this slide, keep in mind that we're exploring how crucial it is to have a clear and well-thought-out response. This moment of anticipation highlights the importance of patience and deliberate thinking when approaching complex questions or decisions. Remember, in the bigger picture, effective responses are often the result of careful reflection, which ultimately leads to better outcomes. So, let's hold on to that idea as we move forward in the presentation. Today, I want to talk about the phospholipid bilayer, which is a fundamental component of cell membranes. This structure is composed of two layers of phospholipids arranged tail to tail. The key point here is that these phospholipids have a hydrophilic, or water-loving, head and hydrophobic, or water-fearing, tails. This arrangement allows the membrane to act as a selective barrier, controlling what enters and exits the cell. The bilayer's fluid nature is crucial for membrane function, enabling things like membrane proteins to move and interact. Overall, the phospholipid bilayer is essential because it provides both protection and flexibility, supporting the cell's overall stability and function within the body. All right, so let's talk about plant food storage polysaccharides. When plants store food, they mainly store energy in the form of complex carbohydrates called polysaccharides. One of the key storage forms is starch, which is a mixture of two molecules, amylose and amylopectin. Starch is highly digestible for humans and many animals, making it an important dietary carbohydrate. Another important storage polysaccharide in plants is glycogen, but it's more common in animals. In plants, starch accumulates in structures like plastids, especially in seeds, tubers, and roots, serving as an energy reserve for later use. Understanding these storage polysaccharides helps us appreciate how plants manage their energy needs and also highlights their significance in our diets and agriculture. So, in essence, starch is the main plant food storage polysaccharide we encounter daily. Answer coming. 5 seconds think. All right, take a moment to gather your thoughts. This slide is about the importance of patience and reflection before jumping to conclusions. Sometimes, the best insights come when we give ourselves a pause to think deeply. It's a reminder that rushing can lead to mistakes, whereas a brief pause allows us to consider all angles. This concept, ties into our overall theme of strategic decision-making and mindful leadership. By integrating patience into our process, we can make more informed and effective choices. So let's remember to pause, reflect, and then act. It's often the key to success. Let's talk about starch. Starch is a carbohydrate that serves as a major energy reserve in plants. When we consume starchy foods like potatoes, rice, or bread, our bodies break down the starch into glucose, which we use for energy. It's important to understand that starch is a complex carbohydrate, meaning it takes longer to digest and provides sustained energy. This makes it a vital part of a balanced diet. In the context of overall nutrition, starch plays a key role in fueling our bodies and maintaining energy levels throughout the day. So, recognizing the significance of starch helps us better understand how different foods contribute to our health and energy needs. Let's explore the question, what is the animal storage polysaccharide? This is an important concept because it ties into how animals store energy efficiently. The key storage form in animals is glycogen, which is a highly branched polysaccharide. Glycogen is mainly stored in the liver and muscles, serving as a readily available energy reserve. This structure allows for quick mobilization of glucose when the body needs it, especially during physical activity or between meals. Understanding glycogen helps us see how animals maintain energy balance and respond to metabolic demands. So, in summary, glycogen is the animal storage polysaccharide that plays a crucial role in energy management. Answer coming soon. Take a moment to think for about 5 seconds. This pause is important because it allows us to process the question or challenge at hand. By giving ourselves a brief moment, we can approach the answer more thoughtfully and clearly. Remember, in our overall discussion, taking a moment to reflect helps us make better decisions and provides more thorough insights. So, let's use these few seconds wisely, and I'll be ready to share the key points once you're ready. Let's take a closer look at glycogen. Glycogen is a vital storage form of glucose in our bodies, primarily found in the liver and muscles. It's like a quick-access energy reserve that our body can draw upon when needed. When blood sugar levels drop or during physical activity, glycogen is broken down into glucose to meet energy demands. Understanding glycogen helps us see how our bodies manage energy efficiently and maintain balance. It ties into the broader theme of how our body stores and utilizes nutrients to keep us functioning optimally. Let's talk about human respiratory pigments. Now, you might be wondering, what exactly are respiratory pigments? Well, these are molecules in our blood that help transport oxygen from the lungs to the tissues and carry carbon dioxide back to the lungs for exhalation. In humans, the primary respiratory pigment is hemoglobin. Hemoglobin is a protein found in red blood cells, and it's what gives our blood its characteristic red color. Its ability to bind oxygen efficiently makes it crucial for our survival. Understanding how hemoglobin functions helps us grasp how our bodies meet their oxygen needs and maintain proper respiratory function. So, when we think about respiratory pigments, hemoglobin is really the key player in humans. Answer coming. 5 seconds think. Now, take a moment to really think about what we've just discussed. This slide is all about pausing to reflect on the key points we've covered so far. It's crucial because sometimes, in our rush to move forward, we miss the deeper understanding. By giving ourselves this brief pause, we can better connect the dots and see how everything fits together. Remember, this reflection step is part of the overall theme of mindful decision-making and continuous learning. So, let's use these few seconds wisely to solidify our grasp on the material before proceeding. Let's take a closer look at hemoglobin. Hemoglobin is a crucial protein found in red blood cells, and its main role is to carry oxygen from the lungs to the tissues throughout the body. It's made up of 4 subunits, each containing a heme group that binds oxygen and a globin protein chain. The ability of hemoglobin to efficiently pick up and release oxygen is vital for maintaining proper tissue oxygenation and overall health. Understanding hemoglobin is central to grasping how our blood functions and how conditions like anemia or certain blood disorders can affect oxygen delivery. Its structure and function are closely tied to the overall theme of blood health and systemic oxygen transport. So, when we talk about blood components, hemoglobin stands out as the key player in ensuring our tissues receive the oxygen they need to function optimally. Let's take a moment to discuss what exactly constitutes a basic blood unit. When we talk about blood units, we're referring to the standardized amount of blood collected for transfusion purposes. Understanding this is crucial because it ensures consistency and safety in blood collection, storage, and transfusion. The typical volume of a basic blood unit can vary slightly depending on the country or organization, but generally, it's around 450 to 500 milliliters. This amount is carefully determined to balance the needs of the patient while maintaining donor safety. Recognizing what makes a blood unit basic helps us appreciate the importance of proper handling and compatibility, which are key themes in blood transfusion practices. So, when you hear about a blood unit, think of it as a well-defined, standardized portion of blood that plays a vital role in medical treatments. Answer coming. 5 seconds think. Take a moment to consider what we've discussed so far. This is a pause to reflect on your understanding. Think about how these concepts connect to the bigger picture we've been exploring. It's important to give yourself time to process before moving forward. Remember, these insights will help you make more informed decisions. So, take a few seconds to internalize the key points before we proceed. Let's take a closer look at the blood cell. Blood cells are essential components of our circulatory system. They play vital roles in transporting oxygen, fighting infections, and maintaining overall health. When we talk about blood cells, we're mainly referring to 3 types: red blood cells, white blood cells, and platelets. Red blood cells are responsible for carrying oxygen from the lungs to the rest of the body and returning carbon dioxide to the lungs for exhalation. White blood cells are the immune system's defenders, protecting us against bacteria, viruses, and other pathogens. Platelets are crucial for blood clotting, helping to stop bleeding when injuries occur. Each type of blood cell has a unique structure and function, but together they work seamlessly to keep us alive and healthy. Understanding blood cells helps us appreciate how our body maintains homeostasis and responds to illness or injury. So let's talk about the largest white blood cell. Have you ever wondered which one stands out the most? Well, the answer is the monocyte. Monocytes are quite impressive because they are the biggest among the white blood cells. They play a crucial role in our immune system, especially in fighting off pathogens. When they leave the bloodstream, they can differentiate into macrophages and help engulf and destroy bacteria, dead cells, and other debris. Recognizing which white blood cell is the largest helps us understand how our immune defenses are structured and how they respond to infections. Remember, the size of these cells often correlates with their function. Monocytes are large because they need to be effective phagocytes. So, in the context of our overall immune system, knowing about monocytes gives us a clearer picture of how the body defends itself. Answer coming. Hourglass not done. 5 seconds to think. Now, as we wait, I want you to consider the importance of this moment. This slide emphasizes patience and reflection, which are critical in our decision-making process. Taking a moment to think allows us to process information more thoroughly, leading to better outcomes. It also ties back to our overall theme of thoughtful analysis and strategic planning. So remember, sometimes the best move is to pause and reflect before acting. This simple step can make a significant difference in achieving success. Let's take a closer look at the monocyte. Monocytes are a type of white blood cell that play a vital role in our immune system. They are part of the body's first line of defense against infection. These cells are produced in the bone marrow and then circulate in the bloodstream. One of their key functions is to act as precursors to macrophages and dendritic cells, which are essential for phagocytosis and antigen presentation. This means they help to identify, engulf, and digest pathogens, and then alert other immune cells to the presence of an invader. Monocytes are also involved in inflammation and tissue repair, making them crucial players in both immune response and healing processes. Understanding monocytes helps us see how our immune system coordinates defense and repair mechanisms throughout the body. So let's talk about where the light reactions of photosynthesis actually take place. This is a key part of understanding how plants convert light energy into chemical energy. The light reactions occur specifically in the thylakoid membranes of the chloroplasts. These are the specialized structures inside plant cells that are packed with chlorophyll, the pigment that captures light. Within these thylakoid membranes, the energy from sunlight excites electrons, kicking off the whole process. This is crucial because it sets the stage for converting light energy into the energy carriers ATP and NADPH, which are then used in the Calvin cycle to produce sugars. So whenever you're asked where the light reactions happen, remember, it's in the thylakoid membranes of the chloroplasts. Recognizing this helps connect the structure of the chloroplast to its function in photosynthesis, which is at the heart of how plants sustain themselves and ultimately support life on Earth. Answer coming. 5 seconds think. All right, take a moment to gather your thoughts. This slide is all about encouraging you to pause and reflect before moving forward. In many situations, especially when making important decisions, a brief pause can really make a difference. It helps us process the information more thoroughly and consider different perspectives. By taking a moment to think, we're more likely to arrive at well-informed, thoughtful conclusions. This concept ties into the overall theme of mindful decision-making and strategic thinking. So remember, sometimes the best action is to pause, reflect, and then proceed confidently. Now let's take a closer look at the thylakoid membrane. This is a crucial structure within chloroplasts where the magic of photosynthesis really happens. The thylakoid membrane is a system of flattened sacs, or disc-like structures, that are stacked to form what's called grana. These membranes are embedded with important proteins and pigments, like chlorophyll, that are essential for capturing light energy. It's within this membrane that the light-dependent reactions occur, converting light energy into chemical energy in the form of ATP and NADPH. These processes are fundamental because they provide the energy needed for the next stage of photosynthesis, the Calvin cycle. So, in essence, the thylakoid membrane is the powerhouse where light energy is initially captured and transformed, setting the stage for the entire process of photosynthesis. So let's talk about where the dark reaction takes place. This part of photosynthesis happens in the stroma of the chloroplast. The stroma is the fluid-filled area surrounding the thylakoid membranes, and it's where the Calvin cycle occurs. This is really important because, unlike the light-dependent reactions that need sunlight, the dark reactions, or light-independent reactions, can proceed without light, but they still require the products of the light reactions, like ATP and NADPH. Understanding the location helps us see how the overall process is organized within the chloroplast, linking the light-dependent and light-independent stages of photosynthesis seamlessly. Answer coming. 5 seconds think. Now, take a moment to reflect on what we've discussed so far. This slide is about the importance of giving yourself time to consider your responses and decisions. In a world that's constantly moving fast, pausing can be a powerful tool to ensure you're making thoughtful choices. It ties into the overall theme of mindful decision-making and strategic thinking. Remember, sometimes the best answers come when you allow yourself that brief moment to think. So, keep this in mind as we move forward. Taking that pause can really make a difference. So now let's talk about the stroma. The stroma is the supportive tissue found in organs like the liver, lungs, and kidneys. It plays a crucial role because it provides the structural framework and nutrients necessary for the function of the surrounding cells. Think of it as the scaffolding and the supply network that keeps everything functioning smoothly. In the context of our overall discussion, understanding the stroma helps us appreciate how tissues maintain their integrity and how diseases, such as cancer, can involve the stromal components. The interactions between the stroma and the parenchymal cells are vital for healthy tissue function, but they can also contribute to pathological processes when they go awry. So, in essence, The stroma is more than just supportive tissue. It's an active participant in tissue health and disease. Recognizing its importance helps us better understand the complexity of organ systems and the potential targets for medical intervention.